Welcome to the third webinar talk in the Innovation and Technology Transfer module under the Future Earth Coast Future Leadership Initiative. So this initiative is supported by Future Earth and in partnership with the UN Ocean Decade Early Career Ocean Professional Program and Ocean KN. And specifically, this webinar series is contributed by the scientists from the Sea Blues and Blue Seas projects. And these two projects together aim to increase blue carbon ecosystem inclusion in national greenhouse gas inventories and reporting. And today we have two talks on ecological processes. So the first talk will be given by Dr. Xiao Li, who is a professor at the Yantai Institute of Coastal Zone Research, Chinese Academic of Sciences. So she will talk about driving mechanisms of greenhouse gas fluxes in Chinese seagrass beds. And the second talk will be delivered by Dr. Steve, a research scientist at the National Center of Scientific Research and Sorbonne University, France. And his talk will show how to combine field observations and laboratory experiments for understanding impacts of multiple environmental stressors on European seagrass ecosystems. So please kindly stay muted during the two talks, and then we will have a Q&A session after these two talks. At then, you can leave your question in the chat or ask directly. Thank you. And then without further ado, yes, Dr. Xiaoli, please feel free to start your presentation. Thank you.
Okay, thank you for the introduction. Hello, everyone. My name is Xiaoli Zhang. Today, I'm very glad to introduce my research here. And my topic is emission fluxes, processes, and driving mechanisms of greenhouse gases, N2O and methane in seagrass beds. First, I briefly introduce these two greenhouse gases. Methane, the second most greenhouse gas after CO2, contributing about 25% of the global warming. Methane has a short atmospheric lifetime of around 12 years, but a very strong warming effect, 28 times of CO2. So, reducing atmospheric methane emission is an effective approach to rapidly mitigate climate change. Atmospheric methane has increased to more than 2.5 times of its pre-industry level, mainly from fossil fuel, agriculture, and wetland emission. N2O is the third most important greenhouse gas, with about 3,000 times of CO2.
Yeah,
N2O has
Xiaoli,
increased.
sorry,
Yes.
the slides is not moving.
No. Okay. No.
Yeah.
Now it's okay.
No, it's not. Okay. Oh, now it's okay. Now it's good. Now it's good.
Oh, okay.
Yeah.
Sorry. Sorry.
Okay. Oh, good.
I can go on.
Yes,
It's okay.
please. Yes.
Okay. N2O has increased by about 25% since the pre-industrial period, and it's mainly from microbial nitrogen cycling. Seagrass is the only flowering plant that can completely live in seawater. Seagrass meadows are one of the typical blue carbon ecosystems. They cover about 0.2% of the global ocean area, but store 10% to 15% of the marine blue carbon. They have multiple ecosystem services, including carbon sequestration, biodiversity support, water quality improvement, coastal protection, nutrient cycling, and fisheries production. As we know, seagrass meadows act as the coastal nitrogen filters with high nitrogen uptake and transformation efficiency. They accumulate large amounts of organic carbon in their sediment, and their anaerobic sediments make ideal environment for methane and NO2 emission. It has been reported that the global seagrass meadows release methane 0.09 to 2.7 tgram per year. But N2O in seagrass sediments shift between weak emission and uptake. It has been reported that methane plus N2O offsets about 30% of the blue carbon in seagrass meadows on a 20-year time scale. But how to produce these greenhouse gases, and what is the individual contributions, and what is the driving factors? We don't know. We know both methane and N2O emission is a balance between production and consumption. In neutral environments, there are three main methane production pathways, including hydrogenol trophic pathway, acyclistic pathway, and mesylotrophic pathways. It has been reported that only mesylotrophic pathway has been found in the seagrass segments. As for the methane oxidation pathways, there are two, including the aerobic pathway and anaerobic pathway, such as sulfate-dependent AOM, demo, and mental-dependent AOM. N2O usually generated from nitrification and incomplete denitrification. During nitrification, ammonia is stepwise oxidized to nitrite, producing N2O as a byproduct, especially in low oxygen environments. During denitrification, N2O acts as an intermediate and high oxygen, limited organic carbon, and elevated sulfide inhibit the final reduction of N2O to nitrogen. Besides, DNRN also produce very little N2O. As for the N2O consumption, complete denitrification is the only biological sink for N2O. It reduces N2O into nitrogen under strictly anaerobic environments. Our research was carried out in Zostera marina and Zostera japonica bears in the northern China. We determined physical chemical factors, including various ecological donors and acceptors. Benthic chambers combined with GC machine was applied to calculate in situ sediment air or sea air fluxes of greenhouse gases. 15 nitrogen stable isotope labeling was used to distinguish the N2O contribution from nitrification, denitrification, and heterotrophic nitrification. This process oxidize organic nitrogen to produce N2O. 13 carbon-labeled substrates were applied to distinguish methane production pathways, but 13 carbon-labeled methane was applied to quantify methane oxidation processes. Further, we use qPCR to quantify the abundance of functional genes. Metagenomics sequencing were further applied to analyze microbial composition. Our first result, N2O south sink processes in seagrass bears. The field investigation indicated that the seawater N2O concentration in seagrass regions is lower than in the bare flats, and the air-sea flux of N2O result indicated that our seagrass bed act as a net sink of N2O. But in the lab incubation experiments, we found that warming and nitrate enrichment significantly enhance the N2O emission in seagrass regions, but has little effect on the bare flat segments. Based on the 15 nitrogen labeled data, we found that in the bare flats, segments, yes, the NG samples, we found the denitrification contributed more than 70% of the N2O production, and the heterotrophic nitrification contributed 28%. But in the seagrass regions, this is the ZJ and ZM regions, the heterotrophic nitrification contributed more than a half of the N2O production, equally even higher than the denitrification. But autotrophic nitrification contributed very little to the N2O production. And based on the specimen correlation analysis, we found that segments, organic carbon, and functional organisms significantly influence N2O production. Further, we conducted metagenomics analysis. Compared with the bare flats, the abundance of genes associated with N2O production, including nitrification and denitrification, were largely downregulated in seagrass sediments. But the abundance of nose gene associated with N2O consumption was largely upregulated. This can explain why the lower N2O contribution in seagrass seawater at the molecular level. Further, we found those two containing microbes may be the main conductor of N2O consumption in the seagrass sediments. Our second result, methane salting processes in seagrass beds. Our field monitoring from August 2023 to August 2024 indicates that sediment air methane fluxes from seagrass region is largely higher than the bare flat regions, and the fluxes is highest in the summer and lowest in winter. PLS pulse model analysis shows that temperature, seagrass vegetation, and nutrients concentrations are the key factors controlling the in situ methane fluxes. And the short-term lab incubation verified that warming clearly enhances methane emission from seagrass beds. Using 13 carbon tracer, the three main methane production pathways were all detected in our seagrass bed. The production rates were higher in seagrass vegetation, in seagrass regions higher in seagrass regions than in the bare flat regions, and the rates were highest in the top sediment layers. Mesotropic pathway is dominant in the methane production. That is red plus blue in the finger. They occupied about more than 80% of the methane production. ST-lystic pathways account for less than 20%. We also found the hydrogenal traffic pathways, but it's contributed very little. The later pathways mainly be found in the seagrass sediments. We also check the methane substrates. This substrate derived from seagrass DOC filtered and metabolized. They can alternatively transfer from the seagrass regions to the bedflat region. So no significant difference were found between the two regions, but the concentrations decrease largely with the increasing depth. From the metagenomics analysis, we also detect all the enzyme of the three methane production pathways. As for the methanogens community, we found methanocyaninases is dominant in the community. They account for over 60%, and they are responsible for the mesotrophic pathway. Besides, we also found some others group of methanogens. For example, some groups reduce CO2 to methane through DIET, that is interspecies electron transfer, and subgroups they can degrade long-chain alkanes to produce methane. Based on our determined methane fluxes and the area of Chinese Zostera marina area, we calculated that the methane emission fluxes in China is very lower than the global average value. For the methane oxidation pathways, we found that the oxidation rate is also higher in seagrass regions than the bedflat regions. In the seagrass sediments, sulfate-dependent AOM and aerobic oxidation pathways are dominant in the seagrass sediments. Especially, we can see the red in the finger means the aerobic oxidation pathways. They can extend down to 37 centimeter later. So this indicated the oxygen loss from the seagrass rhizosphere is very important to the methane oxidations. But in the bedflat sediments, only the sulfate-dependent AOM pathways is dominant. The qPCR result showed that PMOA gene decreased with depth. This gene responsible for aerobic oxidation and nitrate demo pathways. But MCRA genes, the abundance increased with depth. The aerobic methanotropes, these groups, we can see they are enriched in the upper layers, but the anaerobic methanotropes mainly increase with depth. The PLS model analysis showed that the organic matters and seagrass vegetation and microbial abundance were the key factors influence methane oxidation rates. So far, there are very limited data about methane oxidation global. This study we just based on the global area of seagrass beds and the net methane emission rates fluxes global in the global seagrass and our determined methane oxidation data. We calculated that the sedimentary methane oxidation can remove more than 80% of the total in situ produced methane within seagrass ecosystem. So we only just see very low methane emission from seagrass beds. Okay, last, some take-home messages. First, Zostera marina beds in the north of China function as a net sink for N2O, with nitrification and heterotrophic nitrification dominating the in situ N2O production. Two, hydrogenotrophic, acetylastic, and mesotrophic pathways were all found in our seagrass meadows, with the mesotrophic pathway being dominant. The third, the methane production processes is influenced by substrates, seagrass vegetation, and the abundance and the composition of functional microorganisms. Methane oxidation was active in the seagrass meadow, including aerobic oxidation and anaerobic oxidation. The methane oxidation process is mainly regulated by seagrass vegetation and the abundance of functional microorganisms and ecological acceptor. Okay, thank you for your attention.
Yes, thank you, Shirley. And then, yeah, that's a very nice opening for today's first talk. And then now we can switch to our second talk from Steve. And if people have questions, you can already start to type it in the chat if you want, or you can wait till Steve finish his talk and then we will have our QA session. Yeah. So Steve, without further ado,
Yes.
then,
Okay. Okay, that okay now? Can you see my
yeah,
screen?
looks good. Yeah, thanks.
Perfect. Thank you. Good morning, everyone. So today I'm going to talk to you about the activities that we did within the Sea Blues project on what we call the ecological processes, which is mostly to look at the response of blue carbon ecosystem to multiple stressors. So Charlie did a fantastic introduction for me talking about seagrass. So that's fantastic because I'm mostly going to talk to you about seagrass. I want to mention I'm going to talk mostly about the activities that my team has been doing, but I will also talk a little bit about other activities done by people in the Sea Blues Consortium. So feel free if you have questions about those other activities to contact me and I can put you in contact with the good people. Just a brief reminder for the one of you who don't know this project. The Sea Blues project has like three main objectives. The first one is to advance scientific knowledge on areal extent and carbon stocks, sequestration rate, and greenhouse gas fluxes in blue carbon ecosystem. Then we want to provide input to increase inclusion of blue carbon ecosystem in national greenhouse gas inventories. And finally, build partnerships to promote the role of blue carbon for delivering on global climate policy goals. So for that, we're working across Europe. You can see on this map on the right, where we have like different partners all around Europe and access to different sorts of ecosystem, going to microalgal mariculture, macroalgal forest, mostly in the north of Europe, mangroves, mostly in the Caribbean, seagrass with a big effort and the Med Sea on seagrass and tidal marsh. So I will mostly talk about seagrass today. And we have, of course, this very nice collaboration with the Chinese partners with different expertise and access to different ecosystems. So what I'm talking to you about today is mostly the activity we do in the work package 1 in the project. This work package, we have like different objectives. We want to advance the understanding of the distribution and functioning of blue carbon ecosystem, including their climate responses. So how are they going to respond to climate change? And this is mostly what I will talk about today. We want to reduce uncertainties in quantifying emission and sequestration of greenhouse gas in blue carbon ecosystem. And we want to estimate the actual and project potential contribution of greenhouse gas emission and removal by blue carbon ecosystem. Again, looking at the present and to try to project what's going to happen in the future. And the activities that we talk about today are mostly based on the task 1.2 ecological processes, where we want to quantify the ecological processes under different sets of drivers. And for that, we combine field and laboratory experiments in order to mostly look at the changes in the metabolism of the species and the community composing the blue carbon ecosystem. So I'm now going to jump to what we have been doing. For example, I will give you some example about laboratory experiments that we did, and then I will talk about field work too. So the first case study I want to talk about is a flume experiment that we did last year in my lab in Villefranche-sur-Mer. It's in the south of France, close to the border with Italy. And we work during this experiment on Posidonia oceanica. So Posidonia is a seagrass that we have in the Med Sea. It's endemic to the Med Sea, and it's a very, very on storage, because they can make this very thick mat, we call that, where there's this network of roots and sediment that can be several meters thick, and then they can store like large amount of carbon in this mat. What was interesting for us is that the response to multiple drivers of this species is poorly known. And when we look really at what's happening, not only at the individual species, but at the community level, it's virtually unknown. Like we have very few data on that. So what we try to do is to reconstruct those communities in the lab in order to test their response to a combination of drivers. You can see here our experimental facilities, and you can see that what we wanted to do is to recreate inside those flumes, those long aquariums, like artificial communities that really match what we have in the field, with some Posidonia, the bare sediment, as well as the dead mat. Here's like a schematic of what we did. So we expose them in those 3 meters long flumes. We keep them under those conditions for like several months. And we really wanted to mimic the in situ water current, irradiance, temperature in order to really match what we observe in the field. We recreate the natural community, and then we expose them to different conditions of pH and temperature. For temperature, we're really trying to follow the natural daily variation as well as the seasonal variation, so matching what they experience in the field at the same time. For the projections of pH and temperature, we wanted to have like levels expected by the end of the century. And for that, we used the projects. We didn't use model projections, but we have like long time series in the Bay of Villefranche. And we kind of continue the trend that we're observing since 30 years in warming and in decreasing of pH. And we push that towards the end of the century. So we get to warming of about 4 degrees and a decrease of pH of about 0.4 unit for the end of the century. So what we observe, I will present to you, I will talk mostly about the top figures here, where we look at the flux of oxygen. So like net photosynthesis during the day, respiration during the night. And what we can observe here is the photosynthesis as a function of the different treatments. So we have control, low pH treatment, written pH, high temperature, written T, and a combination of low pH, high temperature, the pH plus T. What was very striking was that the net photosynthesis, so primary production of the community, really was stimulated by the low pH condition independently of the temperature. So when we decrease the pH, we increase the primary production. At night, we had something a bit different, where we are mostly an increase in respiration when we are increasing the temperature at higher temperature, but an opposite trend when we had this combined effect of low pH and high temperature. So this was a pretty interesting result. Below that, you have like the flux of DIC, but I will not go in detail into that. But basically for respiration, we have a relatively similar story with like a higher flux of DIC at night in the high temperature. And then during the day, you can see the results are a little bit different than what we observe with oxygen, but this is because there is a combination of the effect of primary production and net calcification on DIC. So what we did then was to combine those results to look at the daily flux that we had in our flumes. And what we observe when we look at the net community primary production is that there was this increase in primary production at low pH, and an even like stronger increase in primary production when we have a combination of high temperature and low pH. In contrast, when we have only an increase of temperature, we have a decrease in primary production, and then the system becomes net heterotroph. For calcification, we observe something kind of really the opposite, where we had like this strong decrease in net calcification at low pH, independently of the temperature. So the system switched to net dissolution, while under ambient condition of pH, we are close to zero, small net calcification in the system. So this implies really those two changes implies like a completely shift in the balance in the ecosystem between net photosynthesis, net primary production. We're going to uptake carbon, and net calcification, where net calcification in contrast emits CO2. So we can estimate that in the future, potentially the meadow would be a better sink of carbon because net primary production will be higher while net calcification will decrease. Another case study, so it's that we have been not only working in the CBRUS project on the metabolisms, we also work on greenhouse gas. So Chiaoli did a very nice talk on this topic, but I just wanted to mention that we also did some experiment. This was mostly done by our colleagues from the CSIC in Spain, so Iris Enriques and our student Emma Orero. So the analyses are still ongoing, so I'm not going to show you a result here, but basically what they did in the lab was to expose those different communities of seagrass to different conditions, again, of temperature, pH, a different range to really try to characterize like the shape of the relationship between methane and CO2 fluxes as a function of like a large range of temperature and pH. So they did that on Zostera in Sweden and on Cimodocea in my lab. So the Cimodocea are another species of seagrass that we find in the Med Sea. Laboratory experiment done within the project where people who have worked on kelps, looking at their response notably to heat waves. This was done mostly by colleagues in Norway. My group did also some work in the Arctic to look at the effect of a large range of temperature and light on the dissolved organic carbon production by Arctic kelp. We also have done some work on Zostera to look at the response to nutrient and light. And colleagues in Turkey has done some very nice experiment in mesocosm, looking at the litter degradation from microphytes and looking at the effect of a large range of salinity and temperature to see how this will affect the degradation of litter from microphytes. Then aside of in addition, I would say in complement to the laboratory experiment, we did also a lot of in situ work because we really like to validate what we observe in the lab with field observation. For that, what we wanted to do was to assess in situ the metabolism of the Posidonia oceanica communities. It's the work done by my group. And for that, we have used two different sort of method. We have used like a gradient flux approach. So the gradient flux is a non-invasive method that allows to determine the metabolisms, so the net primary production, and we can also estimate the net calcification of the community. This is a non-invasive method. We deploy our sensor and basically we measure the gradients of oxygen pH in the water column. We also characterize the water velocity, the water current, and with that we can back calculate what is the primary production and the net calcification of the community. We couple that with benthic chambers in order to really cross-validate the two methods that provide similar results, but a little bit different informations. So here is an example of what we got with this method, with the gradient flux. You can see here you have in pink, you have the oxygen flux that we measured that was in fall, at the end of fall or early winter in the Bay of Villefranche, so near Nice. And what we observe is that the community is mostly net heterotroph, so it's dominated by respiration at this time of the year. can see with all those negative oxygen flux. And in the background, what you see in yellow are the light conditions. So really like just a bit of primary production during the day with the highest light, and then like we switch pretty quickly to net respiration. So then we did that at different season in order to couple that with the variation in environmental parameter, so light, temperature, nutrient in the water column. So then we're going to work on like multi-parameter analysis, of course. So those are very preliminary results because we started this series and it's still ongoing. But what you can see here is that there is this the integrated oxygen flux over a 24-hour daily cycle. So this is the combination of several days of observations. In yellow, you have the light again, and the black bar correspond to the oxygen flux. And what you can see is during fall in November, we are really dominated by respiration with a little bit of primary production. In January, we start to have a bit more primary production. And then when we get to March, in spring, we have a huge increase in primary production. Just pay attention, the scale is not the same here. So we have like a much higher primary production during spring. So not surprising, but we can put some very nice number on that. Another way that we have been trying to test those the response to communities to different drivers in situ is to use what we call natural analogs. In that case, we work in Ischia in Italy. It's a site like near the city of Naples, where there is CO2 bubbling from the seafloor. So there are CO2 vents from volcanic origin. And in this site, we have gradients of pH, so naturally low pH. pH that can be, we have like different sites, but it goes from like pH of about 7 to 7.8 to then our control at pH of about 8. So we have really a pretty nice gradient of pH conditions. Here you can see on these photos how it looks like while we have those bubbles coming from the seafloor. And what we did at this site is we did analysis of the sediment I'll present to you, and then we also did some analysis of the community metabolism. What we observed for the sediment is that there was a very, very clear trend with increasing organic carbon stocks in the sediment when we decrease the pH, going from ambient pH, low pH, so pH of about 7.8 to extreme low, so pH of about 7. We have this pretty clear increase in pH in organic carbon stock. And the same thing with the organic carbon burial rate that increase a lot when we decrease the pH. So some very clean results. So then we wanted to see what can explain that. So we look a little bit at the contribution of the different species to this organic carbon. So using stable isotopes. And what we saw is actually the contribution from the seagrass tend to decrease when we decrease the pH. And this is compensated by an increase of the contribution of the carbon that is stored by the macroalgae and the non-calcifying epiphytes that are actually benefiting from this additional CO2 that can increase their primary production, increase their growth, and we find that in the sediment. So what we do then is to try to combine that with field measurements of metabolism, like similar to what I presented before. And what we see, those are very preliminary results. We just had a few days of deployment, and we'll go back there next week, actually, to do more measurements. But what we see going from the ambient side, where we have respiration, primary production, and then the budget, what we can see is that we have really an increase going from the ambient side to the low pH side, increase in respiration, increase in net primary production. We really have like sort of a faster metabolism when we are in the low pH condition. But this is again to confirm with the next measurements. So just briefly for the in situ work, the other partners from CBlues, there have been also a lot of work that has been done measuring CO2 and methane flux in situ in different environments. So this has been done in Spain on Posidonia oceanica, in France on Cimodocea mido, so there are another kind of seagrass, Sweden on Zostera, and the Caribbean on for mangroves, and in Turkey for salt marshes. So really like a large range of environments were tested. And then we have also worked on assessing the effect of local drivers. For example, the restoration. What is the effect of restoration of seagrass on the CO2 and methane emissions? So here you have an example, for example, of a work done by our Spanish colleagues, where they look at the CO2 flux in meadows that were restored at different time point compared to like a meadow that was non-disturbed originally. We have also done some work on boat anchoring to see at how much the anchoring of boat and the destruction of meadows will affect the capacity of meadow to store carbon. So with this, I will move to the conclusions. So what we saw really here is that we also did some meta-analysis to look at the data that we have on the blue carbon ecosystem and blue carbon ecosystem species. And what we found really is that there is a lack of response, a lack of data on the response of those organisms to multiple drivers. So there are like some disparate studies, but like good studies that really look in depth at the interaction between drivers are really lacking. There is really few studies. What we also saw is that finally complex experiments are necessary, like to recreate community, work on communities. We can really get a different story than when we work on single species. So it's really important to think about like complex experiment to really grasp the processes at the community level. We also saw that the use of natural analogs and seasonal variations are pretty nice options, and it's very complementary to the lab work. The lab work, we can really disentangle, understand the processes, and then we go in the field and we can confirm that. So that's, I think, something that I would really recommend to do. I think then we also saw that it's needed to investigate more driver at the local in the local environments to really adapt our experiment, adapt our studies to what we have locally, because there is we're working with coastal ecosystem with very different impact of local drivers. But I would say in one way I say that, but another way it's also not possible to test all the combination of drivers. So I think it's really important to also work on experiment when we try to understand the processes in order to really understand the response that we observe. And for that, we need to really work a bit more with modelers, work on meta-analysis to really understand the processes behind the responses to multiple drivers. Thank you.
Great. Thanks, Steve. So now is our QA session. And if you want, you can raise your hand by the Zoom function and we'll call your name. And then you can unmute yourself or you can leave your question in the chat box. And now I think there's already a question in the chat. I don't know either Xiaoli or Steve can answer. It's more technical about how to handle the measurement if they cannot do it on site.
I think that's more for Xiaoli.
Yeah, more for Xiaoli.
Okay.
I guess
And
I can also respond. I think it's for carbonate chemistry in general, you don't want to freeze your sample because then you are going to create some decaying of the sample, and you are going to change your DAC and your pH. So I think unfortunately you are not going to get an accurate pH measurement if you freeze your sample.
Yeah, so still try to measure it on site.
Yeah.
Yeah. Cool. And then while we are waiting for maybe other people want to ask question, I have few questions for Steve. I just curious.
Yes.
So when you show the case two in the beginning and for the lab experiment, there's a LED plate on the near the seagrass bed, and then what's the purpose of it?
Uh, which one? Can you repeat the?
Like the first slide you show for the case study two,
Yes.
yeah, with inits, I think. Yeah.
Yeah.
And in the photo you show there's I don't know, there's something shining the light and then yeah.
Actually, I'm not sure. This photo is from here. I think it's just a reflection of the light, no? That
Oh,
is
okay.
on
Okay.
top. Because these photos was from an experiment done in Flums in Sweden. But in there were really like a little bit similar to what I presented for Villefranche. They are trying to reconstruct um, a seagrass meadow, like a seagrass community in flumes,
Hmm.
and then they run incubations with small incubation chambers inside this flume to look at the methane and CO2 flux.
Cool. And then I have second one because the final part that you mentioned, like when it has low pH, then the organic carbon in soil increase and also the burial rate also increase.
Yes,
And then the next slide you mentioned that both photosynthesis and respiration increase.
yes.
And but the NCP decrease, right?
Yeah, I mean, I would be very careful with those results because they are based on like very few measurements we did last year.
Okay.
So that's
Mm-hmm.
why I just mentioned there is something happening with the metabolism of the
Yeah.
community. We
Mm-hmm.
see this sort of like increase in the metabolism, but I will not look too much into the detail about the numbers because those
Yeah.
are very preliminary and we had some issues with sensors, and that's why this year we're going back to do some more in-depth measurements to
And then
confirm
maybe
that
you
and take some
plan
good numbers.
to do it at multiple time points as well.
Yes, yeah, yeah,
Okay,
yeah,
perfect. Yeah.
yeah, that's the goal. And
Cool.
multiple pH too. We have this nice gradient of pH, so ideally we'll have like different pH to look at trends of this.
Yeah. And okay, that's the question I have for Steve. I have one quick question for Xiaoli, I think. Because
Yes.
in on page 15, and then you mentioned the N2O production increase and then N2O consumption, like microbial decrease for seagrass, and then why in seagrass area those community change is because of the environment or is because of seagrass they put some substrate into the yeah what's the mechanism the microbial community change between seagrass and bare soil.
Mm, yes. The seagrass can secrete many organic carbon into sediment, and this organic carbon can be de, 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 degrade
Hmm.
degrade by microbes into some small matters like an acetate and
Hmm.
others.
Ah.
And this substrate can transfer laterally from the seagrass region to the agent bare flats. So usually we can't detect a significant difference of this substrate between
Mm.
these two regions. Yes. But the mm, they can decrease very largely from the surface to the deeper sediments.
Hmm. Okay,
Yes.
nice, thanks. Okay,
Mhm.
so if there's no further, okay, there's one question for Xiaoli. So for 15N stable isotope thing, uh, how do you decide to do incubation for 6 hours? Like why in the end you chose 6 hours for incubation?
Incubation why just 6 hours
Yeah, for
for
15N, yeah.
nitrogen. Yes, yeah, we incubation for longer time, but later the nitrogen the N2O will be uptake by the simulation ecosystem because maybe the oxygen will be consumed, will be exhausted later. Yeah, so the denitrification, the complete denitrification will reduce the N2O2 nitrogen later. Yeah, so I just incubated them for 6 hours. Yeah.
Okay. So like these 6 hours is not how to say the
Uh,
rule. You need to decide yourself. Depends on your temperature, everything, like to see how fast like the process can be completed, right? So the 6 hours maybe will need to be shorter if you do it. Yeah.
Yes, yes. Yeah.
Okay.
If you can put no. I just want to uh, keep the ecosystem like the in situ condition.
Mm.
So we don't disturb the ecosystem. But later the oxygen will be consumed. Yeah. So
Okay.
the nitrogen, yeah, well.
Nice. Okay, perfect. So he got it. Nice. Okay. Then we can officially close today's webinar, and then we thank again for the two presenters, and also thank everyone for connecting. And then the next webinar will be on Fluxes, about in around the end of July-ish. So stay tuned if you want to continue to join and then follow Future Earth Coast, yeah, channel, and then this information will be announced as soon as the date is fixed. Yes. Okay. Perfect.
Thank you.
Thank you.